Good evening, everyone. I'm Kavyansh, and it is my pleasure to welcome you all to the third day of our lecture series on the topic soil-based Gwal Pahari acid. Before we begin, I would like to remind you that if you have any questions, please submit them through the chat box or reserve them until the end of the presentation. It is now my honor to invite Mr. Eswaran sir to deliver today's lecture. Thank you.
Good afternoon to one and all. It's a great opportunity for me to address a gathering through St. Stephen's College, that too the Chemistry Society, a department where I got my first job in 1968, 1st November. You can do the arithmetic. And a joy that I could serve the college for 44 years. In between, of course, I was allowed to go to Deshbandhu College as principal on LIEN, LIEN. So I held my post in St. Stephen's. Today's talk is very specific, but will be a little easier to understand than the previous talks, two talks. This is a paper published by me in 2023, and somebody in this audience has played a very critical role in my post-retirement days to do science, and I acknowledge the presence of one such person who I'm sure will say a few words at the end of the talk, if he is allowed to join. I know that, can we have the slide, the presentation put up? Anur Bhava, did you do that? So the title of this afternoon's talk is Soil-Based Gwal Pahari Acid. Even before I get into the talk proper, I have a few remarks to make. Soil is very important. So this talk is to underline the importance of soil health. And also the fact that if you see Vishwamayin, I don't know whether you read it in any textbook, the name Vishwamayin. And you do realize an Indian touch in that title. Fullar Azirman, the second talk, was the word or the name was invented by me based on fullerene, Ar for aryl, aromatic, Azir for aziridine, and Man for manganese, Fullar Azirman. The title today is Gwal Pahari acid. My science from St. Stephen's College, please remember, St. Stephen's College, I re-emphasize what I said in the last two occasions, is an undergraduate teaching college which has attained excellence and maintained it at almost number one all along these years. Maybe there is some slight change in the recent past, but the college has come back on its track. Students have been encouraged to do science, do other things, excel in whichever area they wish to, be it theatre, be it cricket, be it sports, be it drama, be it music, be it an author. There are many authors, Stephanians, who have written very wonderful books and so on. I don't want to talk about them. Gwal Pahari acid tells you that during my scientific journey, post-retirement, all these lectures cover my work post-retirement. I was in the Regional Center for Biotechnology for 4 years after I had to leave college in 2014 December till 2018 December. But then again I had to switch. This time I had to switch towards Gurugram. There is a place called TERI-Gram. TERI-Gram is part of the structure of the TERI Institutes, the head office in Lodi Road, in India Habitat Center, and there is a Pahari, a small mountain, small hillock. It's called Gwal Pahari. Gwal Pahari is Gwal. The word Gwal could be more or less correlated to the word Gau or Gai. Gwal Pahari. A very interesting place, 2 cricket fields, and there was this TERI-Deakin Nano Biotechnology Centre, a beautiful state-of-the-art laboratory where I was able to once again start working. But I looked around and I said, look, we must work on a team and a certain direction which this entire institute, the TERI institutes were known previously as the Tata Energy Research Institutes. Then the word T stood for the Energy Research Institute, and it was started by the Nobel laureate who won the prize for, that we all of us know for environmental work. And his brother was our own student, St. Stephen's College, who retired as Home Secretary, lives now in Noida. Gwal Pahari acid. None of you could have heard of this term if you had not seen my paper published in 2023. New planets. Who made the planets? In St. Stephen's, we don't only do material science, we also philosophize. We think, we reflect. New planets are not discovered every day. Neither are new amino acids. What are amino acids? Amino acids are these 20 odd amino acids that make up our body, that the peptides, the proteins, along with the nucleotides, DNA, RNA, and what have you, enzymes, etc., your insulin, whatever you think of, proteins, these make up our functional body. They're not discovered every day. I'm announcing the discovery of one more such amino acid today. We don't go to the next slide. We hold on here. What has college taught me in the assembly? Oh, I have been told, let us remember God for a moment. Somebody who says he loves God whom he has not seen. Somebody who says he loves God whom he has not seen, but does not love his brother whom he has seen. He is a liar. वो झूठा है जो बोलता है मैं भगवान को प्यार करता हूं पर जिसको देखा नहीं है, पर अपने भाई को जिसको मैंने देखा है प्यार नहीं करता हूं, तो झूठा है. मेरे को बताया गया था सेंट स्टीफेंस कॉलेज में, इतनी बारी मैंने सुना है, simple prayers, practical prayers. And Stephenians therefore show this ability. Do not go to a congregation. Do not go to a congregation and occupy the most important chair, lest the host comes to you and say, sorry, sir, please go back. Somebody else is very important. He's going to come and occupy this chair. On the other hand, go and occupy an unimportant chair in the congregation. Maybe the host may walk up to you and say, look, you are our most, valued guest. Please come and occupy this chair right in front. These may not look like prayers, but they teach you a certain degree of humility, which St. Stephen's has taught me. These have been very challenging years since 1968 for me, for my science. College has given me everything that I can think of. Cricket, tennis, science, teaching, whatever. But this was an undergraduate college. I want to make and repeat a statement I made in the first 2 days. Colleges are not national laboratories. Colleges are not supposed to be ICERs. Colleges are not IITs. And an undergraduate college must not lose its path. St. Stephen's, the important word is fellowship. What is fellowship? The senior members and the junior members sharing that space with each other and be available for one another throughout the day and night. There are many, many such stories from the history and tradition of St. Stephen's College, to which I will not make a mention just now, but somebody asked me, I will. There it is. What are the questions I'm raising today? You should be clear. At the end of my talk, you can say, sir, you didn't tell us anything about these 3 questions. What are humic substances? Humic, humus. Humic substances are derived from humus. Humus is part of soil. And many such humic materials, let me say humic acids, are known. The question arises, what is Gwal Pahari acid? It is yet another example of a humic acid found in the Gwal Pahari. Can they be called as non-proteinogenic amino acids? Yes, because as far as we understand, these are not important for the makeup of a human body or any form of life on earth. What a limited view. Suppose one such amino acid, and please believe me, many such compounds that I have isolated in the last 5, 7, 10 years are available in huge tonnage quantities on the surface of earth. Maybe they constitute units making life or creating life on some other planet. Let us wait for some more years. The younger people in the audience, will possibly check my statement 20, 30, 50 years from today. That man said these so-called non-proteinogenic amino acids constitute life on such and such part of space or outside space. Non-proteinogenic amino acids. Gwal Pahari acid is one such example. Aha, what a beautiful drawing. I love it. Oh, acha, it was not patented by me. Why? मेरे देश की धरती, मेरे देश की धरती सोना उगले, उगले हो हीरे मोती. I mean, how will you patent your soil of your country यार? कोई देशभक्ति नहीं है आप में क्या? ग्वाल पहाड़ी एसिड जब पानी में डिसॉल्व होता है, when it dissolves in water and you put it in a UV lamp, UV light, it's fluorescent. So beautifully fluorescent. On the right hand side, you see the purple colored test tube. What is that purple color due to? This is due to a nin-hydrogen reaction, very typical of alpha L amino acids, which are the proteinogenic amino acids, which go to making the proteins in our human body. Acha, Gwal Pahari acid is then an amino acid. I am not saying it. I can't see molecules. I told you I can't see nitrogen written on that molecule. I can't see an H2 written there. I can't see a carboxylic acid group written there. I can't see whether they are attached to the same carbon. This is what the organic chemist does day in and day out. He wants precise structures. Biologists want a complete holistic picture. Here, a chemist likes to take apart everything, assess it, and put it back. I hope you understand. On the left-hand side, in the Petri dish is shown Gwal Pahari acid dissolved in water and on which UV light is shining. On the right, a purple color, typical purple color of an alpha L amino acid through the nin-hydrogen reaction. Many students in college do this test in the first year, and they do it again in the third year when you're there doing the compound analysis or in the postgraduate classes. Aha, not patented. You know, organic chemists in general are very, very particular about one thing. They want to know whether it's single molecule or is it a mixture. And the pharmaceutical industry in particular is very keen on molecules which are single, pure, and whose molecular weights can be, as I told you in the case of fluoroazurban, up to the 4th decimal place. Precisely, one can determine the molecular weight. Here, those of you who will read, you read the molecule, the highest peak in this mass spectrum of Gwal Pahari acid is at 577.1. Is that the molecular ion peak? Does it represent the molecular weight? I may clearly express this point, so there is no ambiguity about it. The molecular weight is not 577.1, it is 578.1. Sir, how do you know it? Are you a soothsayer? Are you an astrologer? How did you know what is not there? How are you able to see something? No, we know. Any compound which had an NH bond, the hydrogen in the mass spectrometer will be lost. Aha. This is that hydrogen from one of the NH2 groups. What is the molecular weight of Gwal Pahari acid? The entire audience should note down because very crucial, 578. Of course, one can do a detailed analysis of the mass spectrometer results. We saw a peak at 381.27 in one of the mass spectrum, what is called the Maldi MS spectrum, which I introduced the other day. And it shows fragment peaks. Some chemists, some mass spectrometer people say, no, our instruments give you intact results, so fragments do not take place in the mass spectrometer of some varieties. But this is a fact from the instrument. The 381.27 is a very important observation. Oh, that means it's 380, one hydrogen less. Aha. But we went to another university, Jawaharlal Nehru University. We did another GCMS study, and we got a value of 320. Oh, 320, or is it 319? Those who are studying the Delhi University, many colleges and the university know how to calculate molecular formula. And the molecular formula by the rule of 13. When I was a postgraduate student, this rule did not exist. I'm not going to explain this today. The molecular weight of Gwal Pahari acid 578, using the rule of 13, I can arrive at the molecular formula as C24H38O14N2. It is not a small molecule like carbon dioxide or carbon monoxide, oxalic acid or acetic acid. It's a large molecule. And these come by degradations in solvent. So we have some idea, but there is one problem. Is the molecular weight 578? Then why are you seeing 381 or 380? Why are you seeing 319 or is it 320? If you see 320 and 380, the difference is 60 units, atomic mass units. How many? 60 atomic mass units. But 380 is not the molecular weight. Molecular weight is 578. Oh, I don't understand what is this. That means there is another unit of 198, which adds to 380 to give you 578. I repeat. This unit seen in the mass DMS at 380, M by Z 381 minus 1 is 380, plus a missing figure of 198 adds to 578. Then why did you observe a fragment at 5 to 320? Why did one observe a fragment at 320? Batao bhaiya. 320 or 380 mein antar kitna hai? 60 atomic mass units. Achha, that means we are seeing three parts. The intact molecular weight is, molecule is 578. A fragment is 380. Another fragment is 320. There is a missing fragment which I have not seen. It must be 198. Have I lost my audience? Not yet, I hope. So I must repeat. We are dealing with a molecule of molecular weight 578, never seen anywhere in the world before. It is an amino acid. It is fluorescent. It shows a UV spectrum with very few peaks. There is a peak at 223 nanometers. That means hardly any chromophores. It is somewhat colored, but it's not very deeply colored. I don't know whether we'll show you the color of the sample at all. I forgot. Maybe it's there. Otherwise, we'll show you sometime. As we go and look at the UV spectrum, it tells us it's a bare molecule. इसके पास कोई क्रोमोफोर नहीं, बहुत कम है. आगे जाइए, देखिए इसका FTIR भी देखिए. ऐसे लगता है कि हाथ से ड्रॉ किया है, पर उल्टी तरफ से ड्रॉ किया है. There are peaks around 600, 1692, 1692, 3461. And there are smaller peaks which we are observing. The peak at 1692 is significant. The broad, strong peak at 3461 is important. Please remember, St. Stephen's College is an undergraduate college. I have worked in that college with a small 10 by 10 or is it 8 by 8? I do not know, or 5 by 5 lab all my life. No instruments. तो ये रात को 12 बजे के बाद आते हैं इंस्ट्रूमेंट मेरे घर में. हर जगह मैंने इंडिया में बताया, रात के 12 बजे आते हैं. और there is a story here that the husband and wife, old people, used to make small shoes. How many of you remember the story? तो उसी तरह NMR इंस्ट्रूमेंट भी रात को मेरे घर आ जाता है. मास स्पेक्ट्रोमीटर भी आ जाता है. आज भी, कल भी आया था, चला जाता है 5 बजे. फिर उसके बाद मास स्पेक्ट्रम आ जाता है, स्पेक्ट्रोमीटर, रामन स्पेक्ट्रोमीटर आ जाता है. तो ये क्या बता रहा है कि ये शायद 1692 some carbonyl functionality like carboxylic acid, which is somewhat supported by the 3461 peak, not fully. As we see more spectra, भाई ये ड्रीम कहानी है कि सच कहानी है? Is it a fact or is it fiction? मेरे जो भाषण होते हैं ऐसे लगते हैं फिक्शन है, but they are backed by hard data. Those who do not know what this H1 NMR spectrum shows, or PMR, proton magnetic resonance spectrum shows, it shows that there are no peaks beyond delta 2.2. There is a small hump overlook that hum. Very broad spectrum. But there is one sharp peak, tall peak. It clearly shows for all students, don't worry, you don't need to know NMR spectroscopy. It clearly shows there is no peak at 7 delta on the left-hand side. There is no aromatic ring. Most humic acids described in the world are invariably aromatic in nature because they are formed by the breakdown of lignins in the soil. Ours is either aliphatic or alicyclic, but certainly not aromatic. Now, can you see how many, what was the molecular weight? I told you so many hydrogens, so many carbons. Can we pin down each of those hydrogens? So I will not describe that in detail. I will show you the table. We have done precisely that we can pin down every single hydrogen that is present in the spectrum. We saw the UV spectrum. We saw the FTIR spectrum. We saw the mass spectrum, mass spectra values we saw. Now we assigned the H1 NMR spectrum. In the next slide, we will be seeing something I had to go all the way. A former student of mine, I forgot to send him the link. He worked with the ETH. Those who were asking me questions last time, remember ETH stands for Eidgenössische Technische Hochschule in Zurich. Every single director of that lab has been a Nobel laureate. But they were not, unlike in India, appointed because they won the Nobel Prize. They started from the ranks of the institute as lecturer, assistant lecturer, associate professor, professor, eventually Nobel laureate. So Vipin Kumar, who is working in the Tata Institute of Fundamental Research in Hyderabad, worked in this institute, came back. I went to him 2-3 years ago. The date is mentioned in the spectrum. And we wanted to see whether we can do a solid state NMR spectrum of this. Because we were doing the NMR spectrum in D2O, deuterium oxide. So all the hydrogens, which are acidic, NH2 hydrogen, COOH hydrogen will disappear. So you can see on the left-hand side here, a peak, a broad peak that shows very clearly that the value is somewhere around 180 values mentioned there. So this very clearly shows that this is a carboxylic acid, because we didn't have a proof for a carboxylic acid. Some young students would say, sir, why can't we do it like this? We put bicarbonate solution, there will be effervescence. We are seeing that, but we want instrumental confirmation to us. Next slide shows to you the fact that we have now the same thing tabulated. 18.292, 49.893, 70.493, 173.132 confirms carboxylic acid. The others were also assigned by us as which group. This is a large table. You don't try to strain yourself. But what we have done is we have assigned every single hydrogen in every part of the molecule. Now I'll tell you what I have not told you so far. This molecule consists of 3 parts. One part is very flexible. The other part on the right-hand side, you will see shortly at the end of the talk, is very rigid. In between is a carbon atom to which is attached another carbon atom. You imagine methane. You imagine methane, carbon of methane. There is one OH, there is one CH2OH. And then on the right-hand side and left-hand side are 2 rings. One rigid ring, the other a flexible ring. What does the flexible ring do? It can rotate all the time. It can move around. And since these 2 are attached to a carbon in the middle, the 2 rings can rotate. So the molecule is not, strictly speaking, stable. At the end of the talk, I will ask a question and I will answer it. I do not know whether I should say it now, but let me say it. Is the molecule optically active? Yes, it is chiral. When you have lactic acid with 1 asymmetric carbon atom, 2 raised to power n, 2 raised to power 1, is two isomers, plus and minus. D and L, small d, small l, or capital D, capital L. When you have tartaric acid, you have two chiral centers. 2 raised to power 2 is 4 isomers. You know which isomers I'm talking about, and the work of Louis Pasteur. But in our molecule, wonderful, wonderful. I mean, I was not wanting to tell you, but it has come on the screen. See on the ring A, which is in red color, is multiply substituted. It's a cyclopentane ring, 5 carbon atoms. It shows what is called pseudo-rotation. And there are so many conformations. The ring B, shown in blue, this is the structure of Gwal Pahari acid, is very rigid. The central carbon atom is a carbon atom like a methane carbon atom. It allows the two rings to rotate in clockwise, anti-clockwise, any manner they want to. At the same time, the ring A occupies any number of conformations whose energy differences are very, very minor. That means the molecule can exist in many, many states. I want to ask, how many chiral centers? How many asymmetric carbon atoms? I answer, as many as 13. As many as 13 chiral centers. Oh, 2 raised to power n, 2 raised to power 13 means 8,192 isomers. Tartaric acid, there are only 4 isomers. In hexosis, aldohexosis, to which glucose also belongs, there are 2 raised to power 4, 16 isomers. Here the number of isomers possible is 8,192. Which isomer is ours? What conformation does it occupy? Does it keep rotating? Does it keep changing? The systematic name is, I would rather not read it. It's a very long systematic name. Some computer softwares are able to give if the molecule is not too large. What have I said? We are dealing with a soil-based amino acid with a specific structure determined spectroscopically by mass spectrometry, by UV spectroscopy, by FTIR spectroscopy, Fourier transform infrared spectroscopy, by MALDI-MS. MALDI-MS stands for matrix assisted laser desorption ionization mass spectrometry, MALDI-MS, and we have also on top of it NMR. And what we have not shown you, we will show you, but don't try to understand it. We have caught every single atom in the molecule as per the prediction of a software called ChemDraw. Ours is not a predicted spectrum. Our spectrum is an observed spectrum in the laboratory. We match them and we see one-to-one correspondence. It took a long time for the journal to accept the paper. So the thing is, what does it do? We had our student, I hope whether she's there or not, I don't know. What is the use of studies like this? Can we throw urea out of business? I was told, be careful when you make such statements, because it's a huge lobby, international lobby. Can we do fertilizer-free farming? What you are seeing here is that Gwal Pahari acid brings about tremendous growth of tomatoes. Some of these plants, without even being watered, survived the COVID times when nobody was attending to them. Acha, tomatoes grow much, much better in the presence of small amounts of Gwal Pahari acid. Not at all a surprise here. Suno yaar, Gwal Pahari acid ka दूरी जन्मभूमि कौन सा जन्मभूमि कृष्ण जन्मभूमि से कितना दूर है कोई 100-125 किलोमीटर होगा नहीं 100 to 125 kilometers. Krishna ji was caught, Lord Krishna was caught eating matti. Don't start eating the soil of the matti of St. Stephen's College, but be true to the St. Stephen's College, matti, and the traditions. Don't move away. अरे देखो इसमें दिखाया है कि यदि it is on, the 4 diagrams that you are seeing, whether it's the root length, where is the shoot length, whether it is the, as we say here, both the root length, shoot length, and the fresh weight and the dry weight. You see C, the bar C represents Gwal Pahari acid. There is a scale here. Control T1, T2, T3. T1, T2, T3 are different concentrations of the same Gwal Pahari acid. You see, they are all very much higher. As you keep increasing the concentration, the root length, the shoot length, the fresh weight, dry weight of the plant increases. That means it is a plant growth promotant. Not a surprising thing. It is a natural product. As we show next part, there is a professor, Dr. Saikat Bhattacharya, who collaborated with me in the Regional Center for Biotechnology. His student, Krishnendu Goswami, now in America, postdoctoral work he's doing. He showed that Gwal Pahari acid, this is the plant called Arabidopsis thaliana, a certain genetically modified plant. As I told you the other day, we have, 3 species on which we do a lot of experimental work because their genetics is very close to that of human beings. One of the model plants is Arabidopsis thaliana, which you see on the left-hand side. The one in this photograph, you see the right-hand plant is much larger in size. That has been treated with Gwal Pahari acid. And you can see the control in the black bar compared to the treated sample. The treated sample is much better. When it is, also iron leakage. When iron start leaking from a plant, it is harmful to the plant. But the iron leakage in the presence of Gwal Pahari acid has diminished dramatically. At the same time, the immunity of the plant increases tremendously. I owe this work to Krishnendu Goswami and his professor, Dr. Saikat Bhattacharya, in the Regional Center for Biotechnology, Faridabad. More work was done by us on different areas. I'm not going to explain this. The other model plant that is often used, not plant, model system, is the Drosophila melanogaster, the fruit fly, because it has only a few chromosomes, but its chromosomes are very similar to the human being. Any results that you get from Drosophila is very highly rated. And there are a number of people in India who are very good in this work on Drosophila genetics. This person actually carried out this work and showed, in brief, I will mention, that Drosophila melanogaster likes Gwal Pahari acid, maybe a little shade better than sucrose. कल हमारी लेबोरेटरी में जाके हमारा ग्वाल पहाड़ी एसिड अपने मुंह में डाल लेना, आपको पसंद आए नहीं आए मैं नहीं जानता हूं, पर ड्रोसोफिला तो इसको खुशी से खाता है, पर इससे मरता नहीं है. It is not toxic, but it's not behaving very differently. It is liking it as much as if it were chini, sucrose, and results from, Drosophila melanogaster or the fruit fly. Have you seen the fruit fly? Fruit fly, जो हमारे केले को रखते हैं ना डाइनिंग टेबल पे, छोटे छोटे कीड़े आते हैं उसमें, वो ही फ्रूट फ्लाई. अरे आज का टॉक जब हम खत्म कर रहे हैं, आप सबको मौका मिलेगा सवाल पूछने का. You can ask questions, you can raise your doubts. What I'm trying to tell you is that this is a natural amino acid previously unknown, unidentified. It's a very complicated compound with 13 chiral centers, so 8,192 isomers, optical isomers possible. And these optical isomers will have many, many conformational isomers which differ in their energy by very little amount in kilocalories per mole. So it's very difficult to record an NMR spectrum because NMR spectrum can only study molecules which are static, not changing dramatically. We did one more piece of work which was also not published. We will possibly briefly refer to it. वहां कृष्ण जी का फिर जिक्र आएगा. यदि हम एक एंटी कैंसर ड्रग को इसके साथ जोड़ दें, डॉक्सोरूबिसिन, इस स्लाइड में आपको एक तो ग्वाल पहाड़ी एसिड तो आप समझ ही रहे हैं टॉप में है, उसके साथ हमने डॉक्सोरुबिसिन को जोड़ दिया. Doxorubicin is a very deeply colored drug, red in color. We at some stage make it completely blue and then bring it back to red. This entire sample is red in color. We have done its mass spectrum. We have the results with us. What we have done is we have covalently coupled Gwal Pahari acid, matti, with an anti-cancer drug. What is the hypothesis? If this combination drug covalently bound to each other gets hydrolyzed in the human body more slowly, chances are the well-known side effects of anti-cancer drugs, particularly doxorubicin. I told you the patient does not die of cancer, the patient dies of a heart attack because this is cardiotoxic. We have done some work, but we were not allowed to publish it for some reasons. So I'm not able to claim that it is better than doxorubicin, but we have some data with us. Look, you saw the UV spectrum of Gwal Pahari acid. You saw the UV spectrum of Gwal Pahari acid. देखो आप ये spectrum किस तरह बदल गया. It is going up to high values, up to 500, 534 nanometers, 501 nanometer, 476 nanometers. This is very clearly a fact that is proved that doxorubicin has got attached, covalently attached to our molecule. Acha, that means Krishna ji ne matti kyun khaya? Arre, don't get me wrong. I'm a student of science. I'm not going into mythology. But many children you have seen, other than Krishna ji, also eat matti. Sometimes you may have seen a dog or a cat also eat matti. Please don't start eating matti from tomorrow in St. Stephen's College. They give you good food. अब देखिए, यहाँ हमारा compound on the left-hand side is Gwal Pahari acid. The bar diagram clearly shows that the molecule is hardly cytotoxic. It doesn't do any harm to the human body, normal cells. Similarly, compound D is our Gwal Pahari acid, this time bound to doxorubicin and a concentration at 10 micromole and 20 micromole. It does not show any toxicity. Doxorubicin is so toxic. Can you reduce its toxicity? Can you make it slow release? We have done it. This part was kept out of our publication for some reasons. My student will, maybe someday you share with him, he will tell you why it was kept out, but not for any commercial reasons. Summary of this part is, this is Western blot and immunoblot analysis. Again, you see there are 3 lines here on top. GPA stands for Gwal Pahari acid. GPA doxo stands for the conjugate, and doxo stands for the anti-cancer drug. And then especially the right-hand side, if you see in the different bands that you see, the bands where 20 micromoles of our conjugate is shown, the band is thicker. In all the cases, BAX, caspase 9, cytochrome C with actin, which goes to prove that first, the Gwal Pahari acid, doxorubicin, is not going to kill normal cells. At the same time, it is very effectively killing the conjugate. Gwal Pahari acid, doxorubicin, is killing very effectively cancer cells. The study was done in the lab of my collaborator, who was the senior most professor, Professor Prasanjit, who has been so kind to me. I do not know whether it's in the audience. How many times I should thank him, I do not know. And this result he has shared with us. We remain indebted to him for this help, which would not have happened if I had not spent 4 years in Regional Center for Biotechnology. But if I had not gone to TERI-Deakin Nano Biotechnology Centre, then I would have never picked up working on the soil. Let me leave the slide there. You enjoy this beautiful colored slide. I'll come back to it as we are close to our major talk today, so that questions can be raised at any time after I finish. But before that, a more general point. Does nature take you to these labs? and make you do work in areas which you had never dreamt of. I did not invent the word non-proteinogenic amino acids, but I dream a day when the Mars rover or a rover on Jupiter digs out a little muddy. You know, our own, Chandrayaan dug out some soil on the surface of moon. You know where they did the tests on Earth before they sent this instrument there? They got the soil of Namakkal in Tamil Nadu. Namakkal is the place where Srinivasa Ramanujan, the great mathematician, lived and studied and worked. Then he went to Presidency College, then to Cambridge. But the goddess of Namakkal wrote on the tongue in a dream of Ramanujan. When Ramanujan was in the hospital, because he did not know how one should, cover oneself up in the winter in UK, in England. He had never seen a razai. He had never seen a quilt. So he, through winter, slept on top of the quilt, never covered himself. He took bath, cold water. He cooked his own food. He got tuberculosis. But when his professor, I hope you all remember the name of the professor and the very fantastic meetings about the numbers and, that Ramanujan used to interact with and his various discoveries. The professor, he said, I am an agnostic. I am an agnostic. I don't believe in God. But please, for God's sake, do not disturb this boy. He was barely 28 when he became Fellow of the Royal Society, London. You can't imagine. We are finishing our PhD at 28 or even later in India. It's like a big problem. We're making many policies of education. We are not understanding that St. Stephen's is a unique example, as Dr. V.K.R.V. Rao said in 1981, that UGC should learn from the successful 100-year-old experiment at St. Stephen's College. A college is a place where senior members and junior members live together, learn from each other, and also be available for each other for fellowship. At the same time, they must excel. And St. Stephen's says excel. So we are not in a hurry to interpret the slide. Somebody may explain it on my behalf. I do not mind. But before I conclude, I do want to say very emphatically, let us not lose the path of St. Stephen's College. Sciences came to St. Stephen's in a strict sense only post 1968-70, post 1970. But we have excelled. We have excelled in every field. I said, there is a Stephanian whom I have taught who went to both the North Pole and the South Pole. I can't dare going anywhere near that. And when Sir Edmund Hillary came to college, the student of mine accompanied him to the stage. Sir Edmund Hillary, I heard him saying, he said, in my entire life, I have not learned as much as from this young man. Many good words from him. And then suddenly Sir Edmund Hillary said, I was in the hall. He said, one day I landed in the airport in New Zealand. And a young lady walked up to me and said, sir, are you Sir Edmund Hillary? I said, yes, I am Sir Edmund Hillary. He said, but I read about you in my textbooks in school. I thought you must have been dead long ago. This is said by Sir Edmund Hillary in the hall in St. Stephen's College, which has housed very bare hall. There are no cushions. There were no cushions, I do not know. Bare chairs. Do air conditioners, cushions, sofas make colleges? There's a Nobel laureate from America who said he had a laboratory which was in some backyard of some institute where hardly he had any facilities. But he says a bare room where you can reflect, think creatively is far better than any established laboratory with infrastructures which are now unparalleled. I'm not against instruments. I need access to instruments. Instruments must be accessible to one and all in the country. They have been treated as individual properties by scientists in national labs and so on, as if they have got these instruments with their personal money. Very wrong. It should be discouraged. St. Stephen's does not claim that it has an NMR instrument, that it has a mass spectrometer, that it has a gas chromatography mass spectrometry, it has an LCMS. You go to any university department, they have much more than this. But we have one thing. We have bright students, brilliant students. If they have a role model, if they are a guide, and if they know the college's traditions and live by those traditions, hold on to the truth. अरे सुनो यार, क्या ये केमिस्ट्री की क्लास है, क्या फिलोसफी की क्लास है? कहाँ जा रहे हो जी? अरे सच बात यही है कि लाइफ में रहना है तो सच्चे रास्ते पे जाओ. Truth alone prevails. Don't move away. At the same time, excel in your chosen area of activity. I'm not saying chosen area of chemistry. Any activity that you pick up while you are in college, you learn so many things from each other. There, who's that year which is in kitab likhi hai? What is life? You know, Erwin Schrödinger, who is famous for the Schrödinger equation, which has led to quantum mechanics, wrote the book, What is Life? This came out of a discussion with his friend in the hostel or the residence, as we call it, St. Stephen's, midnight. They were talking. Both were sons of professors. Then he gave lectures in somewhere in England. That became a small book. This is now taught in MA philosophy. What is life? What is life? हम यहाँ किस लिए आए? क्या हमारा ध्येय है? ग्वाल पहाड़ी एक से एक दिन सिखाएं, एक दिन चर्चा करें विश्व महिम पे, एक दिन चर्चा करें किस पे बताओ भैया? सोलर अजिरमान पे. और भी कई टॉपिक्स हैं जो रिटायरमेंट में एक आदमी, अरे साइंस, एक्सपेरिमेंटल साइंस नहीं यार, ये तो कंप्यूटेशनल स्टडी है यार. ये वाला स्लाइड, ये हमारा ग्वाल पहाड़ी एसिड या शायद डॉक्सोरूबिसिन भी लगा हुआ है कि कौन सा वाला है भाई सुजीत? This is a docking of Gwal Pahari acid with the protein which is involved in cancer.
6BHZ protein, anti-cancer protein.
ये तीनों के तीनों शैलेंद्र अस्थाना ने ट्रांसलेशनल हेल्थ साइंस टेक्नोलॉजी इंस्टीट्यूट में हमारे लिए करके दिया है. ये दिखा रहा है that the docking, the way our molecule is gone in, it fits. That is the reason it is active against cancer. It itself, I'm not saying Gwal Pahari acid combined with doxorubicin. नहीं, खुद भी एंटी कैंसर. अच्छा, मट्टी कैंसर खाना, क्या बात कर रहे हैं भैया? मट्टी खाने से कैंसर ठीक हो जाएगा? मैं नहीं कह रहा हूँ, रिजल्ट कह रहा हूँ. पर हो सकता है ये मट्टी थोड़ा जन्मभूमि के पास की मट्टी है. दुनिया में हूँ, दुनिया का तलबगार नहीं हूँ. समझते हो मीनिंग? दुनिया में हूँ, दुनिया का तलबगार नहीं हूँ. बाजार से गुजरा हूँ. Go through the market, maybe window shopping. बाजार से, दुनिया में हूँ, दुनिया का तलबगार नहीं हूँ. बाजार से गुजरा हूँ, खरीदार नहीं हूँ. जिंदा हूँ मगर जीस्त की, I have no desires. I'm alive. जिंदा हूँ मगर जीस्त की लज्जत नहीं बाकी. I have no desires. हर चंद के हूँ. I'm aware of every moment. हर चंद के हूँ, होश में. I'm aware of every moment. होशियार नहीं हूँ, होशियार नहीं हूँ. I'm not clever. वो गुल हूँ किसाने जिसे बर्बाद किया है. उलझूं किसी दामन से मैं वो खार नहीं हूँ. इस खानाए हस्ती से, I'll go through this word. इस खानाए हस्ती से गुजर जाऊंगा बेलौस. I'll go through this world unattached. इस खानाए हस्ती से गुजर जाऊंगा बेलौस. What am I? साया हूं. I'm a shadow. साया हूं फकत नक्श बदीवार नहीं हूं. I'm not a carving on the wall. मैंने कहा था उस दिन, हम लोग तो सेंट स्टीफेंस कॉलेज की दीवारों में चुना दिए गए हैं. We are part of the wall. I carry a certain caricature of a body with me. Just behind me is the Western Ghats and the sun is just set. I know where I am, at what age I am talking to you, but I am devoted to college. I'm committed to my work. I'm committed to science. But if it is just commitment, nobody is going to respect. This is a publication which you have seen is not in a chemistry journal, published in Sustainable Food Systems under the waste management in agro ecosystems. And Gwal Pahari AcSIR is published in Sustainable Development Research in 2023. Co-authors का नाम है, सुजीत अभी बोले थे, कृष्णेंदु अमेरिका में है, साइकत RCB में रीजनल सेंटर में है, अंकित कुमार is joined in industry. That man called Sambashivam Venkat Eswaran is addressing you. And this is Gwal Pahari AcSIR, isolation, characterization of a soil-based plant growth promoting humic acid. It takes a huge amount of effort. I don't think this is not intended to be shown, but since they have put it up, I cannot deny. You see me in the middle. My, as I said, on the left-hand side is my wife. Sujit is to her left, or her right, my left. And there's a Sardarji turbaned gentleman standing. He is a batchmate of somebody who may be present in this audience today. I do not know. We'll check it just now. Gurmeet Singh. He was in the first chemistry honors batch from St. Stephen's College. And just here, he left the post as Vice Chancellor of Pondicherry University. This is my lab, constructed by the people on Gurmeet Singh's left and on Sujit's right. This is a fully equipped laboratory in Pondicherry and with a production unit. God is great. And we have, I don't want to announce it, we may be moving into a lab in Balman, Eswaran, Biomedicine, Material Science. LLP has been converted into a private limited company as demanded by the Foundation of Innovation of Technology Transfer by IIT Delhi. So we are doing it. The next few days, we may get a letter or यार क्या फोटो डाल दिया भैया, ये तो अलाउड ही नहीं है, अच्छा डाल दो यार. ये अब्दुल कलाम जी को मैं 2 बार ही मिला था. जब मेरे को अवार्ड मिला दिल्ली यूनिवर्सिटी में, मैं समझता था आखिर वाले टॉक में दिखाएंगे, पर इन्होंने डाल दिया. अब्दुल कलाम जी ने कॉलेज में मुझसे क्या पूछा बताऊं? कॉलेज में मेरे को इंट्रोड्यूस किया गया तो फाउंडर्स डे में. He asked me in English, what do you teach? You see me smiling when getting the Delhi University Teacher Award, Distinguished Teacher Award. He asked me in English, not in Tamil. He asked me, what research do you do? See, when he meets me in college, he asks me what do you teach? But when he meets me in the university, giving me an award, he asks me, what research do you do? College research is a byproduct. If a teacher teaches in a college, very sincerely, he is available for college. So many jobs I have done. Examinations, student union elections, admissions, running various aspects of college, running as a recording secretary of the governing body, where I'm not supposed to talk. I've spoken to you for an hour today also. I think we are just about an hour, a little less than an hour. I'm not supposed to talk as a recording secretary. I'm only to advise the principal of the college sitting next to him or the chairman. I may at best be allowed to send a small note. Let me tell you, it's midnight, December 4th or 5th, a couple of years ago. A very hot meeting was happening in the governing body. I'm the recording secretary. And Bishop Bederal came out. I was sent away out from the meeting. This is not confidential. I'm not telling anything confidential. So it was so cold. He said, anybody who sits in this room will catch tuberculosis. Then asked the chief election commissioner, Dr. Qureshi, he came to me, what are you writing? I said, sir, I've been sent out to draft something. I cannot tell you what I'm writing. So he said, no, I'm going for a party at midnight. So he left. Bishop Bederal died within a year. I came back to the meeting to read the minutes. I cannot tell you what I read in the meeting. Very difficult sentences I had to read. But these are the jobs I have done for college. played cricket, played tennis, played and done everything that you can imagine. But we were available round the clock for our students. And many of our students who may not be present today may listen to it someday. I remain grateful to them because they challenged me as first year, second year students. All have come to college, sorry, except a few. All have come because they could not make it to the IIT. They came in a somewhat depressed state, but they found college such a wonderful place. I can give you a talk for the next 2 hours on the brilliant students who have passed out of this Department of Chemistry, of the science block, and in general from St. Stephen's College. College is great. Let us work for the college. Let us not try to convert it into a pseudo research laboratory with publications of impact factor journals which are above 4. One lady who was the director of PHSTI told us, I was in the audience, she said, I will consider only those papers which have an impact factor above 4. Others are junk. This is okay for the national labs where the government is spending huge amounts of money. What is the money that is being spent on research? We did not have money for 40, 50 years. We did not have money for even buying chemicals in St. Stephen's College. Such was the amount that was granted. Money did not make college great. It is a dedication of teachers. It is not whether they are PhDs or they are what degrees they hold, whether they have done work in Oxford or Cambridge or Harvard or MIT, is the dedication. I have mentioned the other name, one name of Professor Muhammad Amin. There are so many stories about him. He did not do a PhD, but he was an LLB, and he worked in Cornell for 3 years. Let me mention one name more today. Professor N.C. Ray. There is an N.C. Ray Memorial talk and so on in the Economic Society. He is not a PhD, but he interacted in the Delhi School of Economics at an equal level with Amartya Sen. The world knows Amartya Sen, does not know N.C. Ray. People decided which whether it was in Delhi by looking at the dress that he was wearing. Such was his, custom, and so popular he was. He built up the economics department, which some people think is one of the best departments in the country. Of course, they moved from there to Delhi School of Economics or to JNU or to London School of Economics. हम लोग अपने आप को किसी एक ढांचे में नहीं रखते हैं. केमिस्ट्री है तो गाना नहीं गाना है, क्रिकेट नहीं खेलना है. Arun Lal serves me. Kirti Azad, I have been his advisor. And so many other cricketers. Over the other day, I spoke to another IAS from Chennai, who spoke to me, a former Michael Dalvi, is a Tamilian. And I called him the palm tree heater. भाई सुनो, आज का भाषण खत्म होना चाहिए ना, डिनर तो नहीं सर्व कर रहे हैं आप. So I think I should bring myself to a close. I thank all of you for your patience, but I must conclude. एक बात बताइए, क्या काम बाकी रह गया? क्या सचमुच में ग्वाल पहाड़ी ऐसे कोई मजेदार चीज है? Can it be combined with an anti-cancer drug? It is not toxic. मट्टी से क्या कोई दवाई बनती है? मैं बाबा रामदेव नहीं हूँ, मैं तो ऑर्गेनिक केमिस्ट हूँ. अंडर ग्रेजुएट बच्चों को पढ़ाया, यूनिवर्सिटी में पोस्ट ग्रेजुएट बच्चों को पढ़ाया, MSc, MTech, PhD कराई. 10 से ज्यादा PhDs मैंने कराई हैं. मेरे हाथ में आज भी एक बच्ची आई, सर आपके पास तो कुछ भी नहीं है. मैंने कहा मेरे पास दिल है, मैं दिन रात काम करूंगा, मेरे को कोई रोक नहीं सकता. आप में से भी मेरे से यही प्रेरणा ले. मेरी उम्र आप जानते हैं, सन 2012 में मैं 65 साल की उम्र में रिटायर कर चुका हूँ. आप देख रहे हैं 1 घंटे से ऊपर, तकरीबन 1 घंटा मैं आपके साथ बात कर रहा हूँ. एक बूंद पानी भी नहीं पिया. पर मैं चाहता हूँ मेरे भाषण को सुन के, मेरे टॉक को सुन के हमारे रोंगटे खड़े हो जाए, गूस बम्प्स हमारे. और हम चाहें कि अपनी जिंदगी में हम भी ऐसा करें. मुश्किल, डिफिकल्ट पाथ चूज करें, not the easiest path. I wish each and every participant, I do not know you by name, I have not tried to see who is present, who all are present, but I am sure Sujit and others will educate me, our administrators will educate me. There are some people in the audience who need to be noted and acknowledge their presence. I will do so.
Yes, yes, sir. Ajit sir is in the meeting, I think.
can we ask him to allow his mic to be switched on and his video to be, the video to be switched on?
Yes, sir. Can I request to sir? Okay, sir is just.
Yeah, okay. so kind of you,
Hold on.
Doctor.
Let me introduce.
But this is your. I don't want to really appear on the screen because I'm here to listen to you. Fascinating.
आदित्य सुनो यार, मैं आपको जानता हूँ, हाथ जोड़ता हूँ, अपना चेहरा तो बच्चों को दिखा दो। मैं बोलूं फिर आप बोलना। Ajit Seth was in college for his undergraduate 1970-73, a batch of students who are alive. They are all closely in touch with me. So many of their batchmates. He passed into the IAS and served in the UP cadre, and he did so many wonderful things that he became Cabinet Secretary, Government of India, into the first phase of the Narendra Modi tenure in post 2014. He continued to be Cabinet Secretary, but he was given extension. He is a highly respected civil servant. But एक बात याद रखो, सेंट स्टीफेंस वाले, मैं किसी को कह रहा था, भाई शिव जी बहुत भोले होते हैं, शिव जी को प्लीज करना बहुत आसान होता है, पर एक बात याद रखिए, गलती करें तो शिव जी आपको पनिशमेंट जरूर देते, पर आप अच्छा काम करें तो दुनिया आपकी दाद कर आपको मांगती है, और अजीत सेठ ने यही सब किया है। मैं चाहूंगा कि यदि वो कुछ शब्द बोले, आप वीडियो में आए तो बेहतर है, 2-4 शब्द बोले। बहुत अपने आप को दबा के रखते हैं, बहुत लो प्रोफाइल रहते हैं, तो ये स्टीफेनियन का स्टाइल है। अजीत कुछ तो बोलो यार, बच्चों को समझाओ।
इनफैक्ट मैं कोशिश कर रहा हूँ कि आई गेट ऑन टू द वीडियो, बट माय फोन इज नॉट अलाउइंग इट फॉर व्हाटएवर रीजन, आई डोंट नो। लाइक तो हो गया, बट आई कीप प्रेसिंग इट, मे बी सुजीत विल एक्सप्लेन टू मी one day. But no, I this is the first of your talks that I have joined in, and I must say that I hope there is a recording available somewhere, because this is a talk I would like to share with some others I love, and this is it transports me back to college, and I find myself sitting back in the classrooms there, and listening to the what you are saying now. I think you used to say in that vein in the past also. And this is education. I mean, it is not textbooks, or it's not merely textbooks. So this is something which we are proud of, and we all think about it all the time, and it's become a part of us. And I think thanks to you and to some others like you, that I don't know, I have no really words to express, but you know what I feel about you and what I feel about people like you. So, but what you have said is absolutely right. I mean, it's not the desks and the rooms that make a college. It is the software there, the human beings who, and the students also. That together, the chemistry of the students and the teachers. That
most important.
So that's all. I mean, I don't know how to put it, but this has been and I'm glad that you have deviated from pure chemistry or pure purely the work that you've been doing, but a few sides on your learnings in life. This is what makes for good teaching. So this is something which I cherish, and I think all others like me would do that. I won't say anything else.
Thank you, Ajit, for coming and being on the in the session, and also for your kind words. Now the administrators, the organizers should take over. If there are any questions, I'm available.
Thank you so much, sir. People, if you have any questions, please feel free to ask. You can write it down on the chat box or ask sir by opening your mic.
I have one question, Saptarshi. So basically, what is multi? Why are we using multi? Can we use multi in order to depict silver nanoparticles?
देखो सिल्वर का तो पता नहीं, you have to read. But Maldi was introduced because I told you the other day in my second lecture, I presume, that if you want to find out the molecular weight of insulin to the fourth decimal place, it was impossible previously. We only calculated based on the amino acids which are going in and so on. It's very difficult to precisely determine the molecular weight of insulin up to the fourth decimal place. When you throw a laser at the molecule, a small part of that molecule is coming into the mass spectrometer, very small part. And the instrument is so sensitive. We are not vaporizing insulin. By throwing that laser of a particular intensity and wavelength, small part of the protein, large protein, insulin, is carried into the mass spectrometer. I am not an instrumental scientist to explain more, but the instrument is able to precisely determine the molecular weight of the intact molecule without the molecule fragmenting. You know what is insulin? Two chains
Yes.
by three disulfide bonds. If the molecule breaks, the disulfide bonds are very, very sensitive bonds. But without any cleavage, without any breakdown, without any fragmentation, the molecule is studied intact. So you can get the molecular weight to the fourth decimal place. I never imagined that I will ever get access to such a facility in St. Stephen's College. But God took me to different places. I worked abroad in many places, but I love my country. I love college. I like to be with my people. I occasionally go to do science at the highest places in the world, but do not be selfish.
Another question, sir. While we see nanoparticles, there is one very useful nanoparticles that is called gold nanoparticles. You might have heard of. So basically, they are used of visible wavelength matrix. So can we use it? Can we use this gold nanoparticles for anti-cancer treatment by using multi-technique, any kind of?
I am happy. At the same time, I want to suggest to you that while I remember somebody predicted in the 60s, it was that author of physics who became a Nobel laureate subsequently. He said there is much room at the bottom. What he meant was that nanotechnology can be developed. But more importantly, what you are saying is those who have seen nanoparticles, gold. What is the color of gold supposed to be? Shining yellow gold. But when you make nanoparticles, it has different colors, red, I don't know what all, gold. So the particle size is very important, but the activity increases tremendously. But gold is a very difficult element to treat human beings with. I know somebody related to me. He was in the army cricket team of the services. So he had pain in his knee. So he went before his annual medical checkup. He went and asked somebody to give him a gold bhasma. He ate that bhasma. The entire immune system became zero. He was admitted from the forward areas. He was flown to Chandigarh. From Chandigarh, he was flown to Delhi, and he died in 1989 in the army hospital in Delhi in the cantonment because of gold poisoning. Gold has to be given to the human body in a very, very controlled manner because it can be highly toxic. When it is a nanoparticle, it can be in very small amounts, but still monitoring is very important. We are trying to treat cancer, but as I said, doxorubicin affects the human heart. You know what other side effects are there with chemotherapy. I don't want to describe how costly it is. A family where one patient of cancer is there, the entire family gets destroyed. They become, without any funds, they become very poor because the anti-cancer drugs are very costly and very toxic. So gold nanoparticles सुनना बहुत अच्छा लग रहा है, कलर बहुत सुंदर होते हैं.
Sir, one thing I would like to add: if the gold nanoparticle size is 20 nanometer, color is red.
चर्चा ये नहीं है, चर्चा ये मैं मानता हूँ ऑरेंज है, रेड है, पर चर्चा ये है कि क्या इसको कैंसर में ट्रीटमेंट में बहुत लोग कोशिश कर रहे हैं, पर हमें पता है टॉक्सिसिटी वर्सेस एक्टिविटी। अब हमारे एक आगे तो इसको ही बैलेंस कर। और अदर एनी अदर क्वेश्चन?
Sir, another question from my side. Gold nanoparticles are used in verticillium species.
हाँ,
Sir, why are we using verticillium species? Means what is the effect of gold nanoparticles on verticillium species?
will you describe to our audience what is this verticillium species?
Sir,
Please
basically
describe.
verticillium. First of all, verticillium species, it's a part of microbial nanotechnology. First, microbial nanotechnology is a combination of microbiology as well as nanotechnology. So this verticillium species is a fungi, that is verticillium verticillium is basically fungi. So we can see that they are used in various types of crop. So from that crop, if we take the crop, we can find that what kind of toxin is there. We can find that there are crude toxins such as toxin 3450, toxin VP28. So basically they exhibit toxicity on we.
ये हमारा
Yes.
ग्वाल पहाड़ी एसिड का जीटा पोटेंशियल कितना है? क्या साइज?
Yes, sir, I can.
Size is almost 12 nanometer, 12 or 13 nanometer.
30 nanometers. Even Gwal Pahari acid is a nano material, ours. So you are very much interested in nanotechnology.
Rohan Kumar has asked in the chat that since GPA is not observed in the organic life of Earth, so do you have any way to understand its working in the extraterrestrial life?
बहुत अच्छा सवाल है। कौन बोल रहे हैं? बहुत अच्छा सवाल है। कौन है?
Rohan Kumar.
रोहन कुमार, my student Manurभाव कहाँ है? Manurभाव, please switch on your mic. You can
Yes, sir.
So,
Yes, sir. Yes, sir.
he has recorded a large, अपना वीडियो भी चला लो. He has recorded a large number of Raman spectra of many non-proteinogenic amino acids. And I have defined to him what is called a signature peak. बड़ा identifiable peak है. मैंने कहा हमारे कंपाउंड का आप रोवर में, चंद्रयान में, रोवर जो मार्स में गया है नासा के पास से, उसमें डाटा में कंप्यूटर में डाल दीजिए, और यदि ऐसा एक पीक 920 सेंटीमीटर इनवर्स पे या 1050 पे जो हमने देखे हैं वो वहां नजर आ जाए तो आपको पता चला है कि ये वही वाला है जो ग्वाल पहाड़ी में हमने देखा, या हमने और अमीनो एसिड कहीं नैवेली में देखा, वहां से हमने देखा। ये डाटा हमारा आप डाल दीजिए। मनोभाव, आप कुछ देर इस पे बोले ना, ये हमारा सवाल समझते हैं। सवाल क्या है? वो कह
Yes,
रहे
sir. Yes, sir. He has asked that since GPA is not observed in the organic life of Earth, do you have any way to understand its working in extraterrestrial life?
Yes.
And if we have to,
We like this question.
this question is very deep in a sense and also very complicated to answer. To understand its working in extraterrestrial life, also we have to know about what kind of working we want to know in extraterrestrial life. The proposition which Sir is asserting is that GPA or such non-proteinogenic amino acids could serve as basic fundamental elements for a new form of life. We all know about the dogma of molecular biology, how from DNA we go to RNA, and from RNA by translation we go to proteins. But we are aware of only the standard 20 amino acids protein synthesis, which we are aware of, and we are not aware of how possibly new types of peptides, proteins, which could be uh, basic elements for exolives. So what we are planning is that we recorded Raman spectra for these molecules, and we got signature peaks for these molecules. And if we are sending these molecules, and we did try to ISRO and also European Union, their organization, space organization, that they keep this data in their rovers, and when they are doing their investigations on other planets, they could possibly find such a new non-proteinogenic amino acids. So this is only a hypothesis which is to be explored further. I hope that it answers some of the part Mr. Rohan has asked, although it is a complicated question. We may have to discuss it further.
Thank you, Manur Bhav. You can switch off and switch off your mic. Very kind of you. He gave a very good, clear answer. Next question, if any.
Rohan Kumar has asked this question: while searching for GPA-based life is interesting, have you tried to make your own GPA organism in biolab?
See, I am not a man who, I'm not, what do you call it, that, that man who touched everything became gold. Midas touch. I can't synthesize life, but I predict that these are amino acids. We saw the structure. There are 2 alpha amino acid units there on each side. We have made, in many cases, many such compounds. This is not just one of the compounds that I mentioned today. Many such compounds. One such compound we have made in 3.5 kilograms. Another, this July, Sujit made it. Dr. Sujit made it in 100 grams. We can upscale it to kilograms. We may not make species, organisms, but if we can use any of them for treating cancer, for treating Alzheimer's, for treating Parkinson's, for treating dementia, then I will be very happy. We have huge amounts of these previously unknown, non-proteinogenic amino acids. But I don't imagine that I have the ability to constitute life and make an organism of my own in the laboratory. But I leave it to the audience and the young people to use these amino acids. We can give you any amount of these amino acids. We have distributed it in many places in the country. And I'm hoping after 11 years, after 2013 years, 2011 was my last visit abroad to United States. I toured the whole of United States, lectured in many places. After that, I was denied a visa since 2011. But now I've been given a visa for 10 years because they think I'm harmless. But they did not know that I'm not doing any trade with them or competing with them. They have their own. You'll hear me in one of these talks now, but I don't want to digress or move away. But I want to say that I want to announce many of these results which you are hearing. The ACS, American Chemical Society 2025 March meeting. But it means that I spend airfare for the 2 of us. For touring in different places, I have to stay, money for stay. I need to have some money for food, and then I lecture. American Chemical Society meeting, spring meeting in 2025 in San Diego. Looking forward, because very few people have heard me in this country on these topics. Because I was literally in Agyatwas. Those who understand what is Agyatwas. For 14 years, you are banished to the forest. I was living with monkeys in Faridabad. They would come when I sit for lunch. They would come and pound my roof. Say, let me in, I will eat with you your lunch. So, but this Agyatwas period has made me produce so much. I hope people in the country will hear me, our own former students who have no idea what I'm doing. I don't blame them, because one scientist who is so kind to me told me in Faridabad, sir, ये सब आप करते कैसे हैं? हमारा तो जिंदगी का एक दिन भी काटना इतना मुश्किल है, घर का काम है, बच्चे हैं, सब कुछ है, दुनियादारी है, तो हम कैसे काम करें? आप फिर किताबें भी लिखते हैं, भाषण भी देते हैं, और सिर्फ ये काम नहीं, हर संडे 17 साल I have done without any interruption into this year, Vivekanand Youth Forum in the Ramakrishna Mission. There are a large number of youth who are associated with me, who are prepared to serve the country. They're totally selfless. Any other question or comments? We should slowly bring this meeting to a close. कोई है chat box में questions? पढ़िए. I think question क्या है भाई?
Yes, there is a question. I think we can use molecular dynamics simulations to simulate how the acid behaves under different conditions, like varying temperatures, atmospheric compositions, and then scientists consider alternative biochemistries like silicon-based life forms. So we can do it using MD simulations, I think.
कौन है ये फ्यूचरिस्टिक
Tejasi Gupta. Tejasi
है
Gupta. Tejasi
कहाँ
Gupta.
तेजस्वी कहाँ से हैं भैया बहुत अच्छा सवाल है मैं अभी इसके ऊपर कुछ कमेंट करूंगा कहाँ से हैं तेजस्वी
Sir, St. Stephen's College.
हैं which year
Third year.
wonderful you see your question is very pregnant I hope you allow me to use the word very pregnant with possibilities today I have said you need a lab you need chemicals you need money you need students but if you have good computer and you are very good chem informatics or for that matter using softwares or generate software and do all this body dimension computational studies and we have one or two faculty in college who are very good in it. I do not know whether they have attended this. I mentioned Raki Tareja, who's a gold medalist from Delhi University MSc. That's why I took in, but she's more interested in NCC. But she's very good. There are others in the university, there are others in the country all over who are very good in computational studies. Your prediction that we could do all this possibility instead of carbon using silicon for a life, instead of alanine or say tyrosine or phenylalanine, these or say tryptophan, which is so important to the human body, could we not? But I want you to note what you have heard from me is only one such amino acid, non-proteinogenic. We have a range of them with us, and some of them in kilogram quantities. But what you are saying is very good. In addition, we could do computational studies. Dr. Shalid Rastana in THSTI has been helping us with this. Dr. I think Goswami, I forget his full name, from the Central Salt Research Marine Institute, who the director was our Pushpito Roy, 1974 batch from St. Stephen's College. In that lab, there is a computational chemist who helped us. Christopher Haddad from the Ohio State University. I published with him on computational work. So I forget the scientist from Bhavnagar. क्या नाम है भाई मैं भूल रहा हूँ भावनगर से CSIR lab.
विश्वजीत गांगुली.
विश्वजीत नाम क्या नाम
विश्वजीत गांगुली. विश्वजीत गांगुली.
With CNR Rao, there is another scientist with whom our students have done some summer or civil internship. He's very good. There are a lot of people in the country in different labs who are computational chemists. There are people abroad, including the Nobel laureate, Roald A. Hoffman. You could work with them. And yes, answer your own question. Is it possible to simulate life using such non-proteinogenic amino acids? Your question encourages me to make a different statement. You see, there are some reactions in organic chemistry which are called no reaction mechanism, no mechanism reactions. No mechanism reactions. Then came Woodward, Robert Burns Woodward, who was already a Nobel laureate, walked up to the laboratory at the office of Roald A. Hoffman, a young scientist, a young faculty at Harvard. And Roald Hoffman writes such wonderful books which are so readable. He says, I was in a flight and my wife was sitting to my side and she asked me, what are you drawing? I said, I'm drawing the structure of a beautiful molecule. He said, what? How can a molecule be beautiful? He writes essays. He's a computational chemist. He and Woodward put up the Woodward-Hoffman rules. By that time, Woodward had died, so he could not share the Nobel Prize. Roald Hoffman got the Nobel Prize with two other scientists. What did they do? They showed these reactions to be pericyclic reactions. They were not no mechanism reactions. They were orbital symmetry rules based on quantum mechanics, which they put up and all these reactions called electrocyclic reactions, cycloaddition reactions, sigmatropic shifts are all now part of undergraduate and postgraduate textbooks. That was 1967-68. So it is certainly possible, there are some parts of DNA which were mentioned as no role. They have no role, right? And then later people discovered that they have a very important role in entire biochemistry and biology. So when we are ignorant, now these amino acids are present, these non-proteinogenic amino acids are present in tonnage quantities on the surface of Earth. And we are saying they have no use. We are saying so because of our ignorance. There's nothing wrong with these compounds. We do not know. One of these I will possibly mention in one of my lectures in the series, which we have made 3.5 kilograms from 10 kilograms in the Pondicherry plant. Believe me, we burn it in our power plants. Lignite, Neyveli lignite, we burn because we do not know how to use it. Nobody in the world, underline, nobody in the world has isolated the kind of compounds that we have identified, isolated in large amounts, put up their structure, studied their use in material science, in biology, in biomedicine, and so on. They are with me in huge amounts. But I wonder if this young lady from St. Stephen's third year will help us do computational studies on these molecules and see, as the other young man asked, can we do constitute life using not alanine, glycine, tyrosine, tryptophan. I don't want to name all the 21, the different 7 types, but can we make constitute life? It's, I think, one of the most wonderful questions that was asked today. Yes, someday I think nature does not waste anything. Nature has produced these compounds in tonnage quantities all over the surface of Earth. We do not know what is its use. And maybe there is some, organism below the surface of Earth, maybe termites, I don't know what, various kinds of organisms that live and thrive under the soil or in the soil, keep it very, very safe. That very highly respected, especially young people love him. He drives the motorcycle, he rides the motorcycle, he rides the motorcycle from London to India, and his center, spiritual center in not very far from where I'm talking to you from. I hope you know the name. And there are so many things to discover in this world. We close our eyes to. हमें पता ही नहीं हो सकता ये फॉर्म ऑफ लाइफ एक रोडेंट हो जो सरफेस के नीचे है वो लिग्नाइट कोयले को बना रहा है वो कोयला काले रंग का है कोई यूज नहीं है उसको जला देते हैं पोल्यूट करते हैं एटमॉस्फेयर को 2030 में इंडिया में 2050 में उसका बर्न करना अलाउड नहीं है we are not going to allow power generation using coal of any form one or the other and pollute. Therefore your questions are very pregnant. All of you have asked so many possibilities. I do not know whether I can do all that work. So this work is handed over to young people in this audience, especially from St. Stephen's College or any other institution who wish to join us and help us. Thank you so much. Are we close to the end? Yeah. Tejasvi Gupta. Kabhi milna mein baat karenge. Yes, my young friend. Is it all for the day?
Yes, sir. Thank you everyone for joining us on the third day of our lecture series. We will be sharing feedback forms shortly. Please take a moment to provide your invaluable insights. Once again, I would like to express my gratitude to Dr. Eswaran for his informative and engaging lecture today. Thank you so much, sir.
Thank you. Delighted. Till we meet again. Thank you. I'm leaving the meeting. Bye.